to the second Learn debate. This is the second because there was a previous edition last year on group selection. As Sandra said, I'm Max Reuter. I have nothing to do with Learn, but as a director of the Center of Ecology and Evolution, so maybe part of the distant family, so I've been asked to introduce this event. Okay, so we will talk about epigenetics today, and in particular we want to ask the question whether the increasing knowledge that we have of epigenetic effects and how they work requires us to update evolutionary theory to take into account evolutionary change that is not driven by sequence change, but by changes in, well, other things. Epigenetic changes. Okay, I've been told that the main purpose of my intervention is to draw your attention to this schedule. So we will have a 20-minute presentation by each of the four speakers. Then we will have a tea break, and then we will have 10 minutes of rebuttal from each of the speakers. After which you can ask questions. We will vote, and then we will become friends again over a glass of wine in the end. Okay? Also, I would like to point your attention to one poster which is put up there, which deals with the role of culture in evolutionary change and the equivalence between culture and epigenetics. Is that right? Okay, good. All right. So the term epigenetics was coined by Conrad Waddington, who was interested in development and developmental genetics. And epigenetics is the study of, I got this from Wikipedia this morning, inherited phenotypic changes which are not caused by mutations in the DNA sequence, but by some other modification of the DNA. Okay? There are several mechanisms in which how DNA can be modified to cause epigenetic effects. So there are changes or modifications in the chromatin structure, so the secondary and tertiary structure of DNA, how it is wound around histones and so on, and wrapped up in the cell. So there are effects like acetylation, methylation, ubiquitination, phosphorylation, and so forth. And there are also changes to the DNA molecule itself, as for example in DNA methylation. And all these changes will have an effect on the way how genes are expressed, and that of course has then knock-on effects on the phenotype. Okay? These different modifications of the chromatin and the DNA can be transmitted through cell divisions and also through generations. Transmission through cell divisions is very important in development of our body, for example. So you have the liver, for example, developing from cells that are differentiated as liver cells. And they are differentiated, and they will transmit this differentiated state to their offspring, if you want, when they divide. Okay, so this type of differentiation is often reset when you create a new generation of offspring, well, in the gametes. But there are also instances where epigenetic changes are transmitted between generations. So, for example, you have genetic imprinting, which cause parent of origin is the term. For example, cause maternal and paternal alleles to be differently expressed. So that is something where some modification has been transmitted between generations. And in organisms that don't have a germline, you can also have environmentally caused modifications which are transmitted between generations. And this, of course, this is where Lamarr comes in, because you can have an effect of the environment on the individual, which is transmitted through subsequent generations. Okay, which is something which is not possible with random mutation in the which is accounted for in traditional evolutionary theory. Okay, so we want to debate whether epigenetic effects require us to update our evolutionary theory in order to account for the effect that these modifications can have on the prediction of the direction of evolutionary change and also the rate of evolutionary change. Okay, so we have 4 speakers who will debate this question. I go through them in alphabetical order just to be completely unbiased. Okay, the first in alphabetical order is Vincent Collot, who is a researcher at the National Center for Scientific Research in France. He is a directeur de recherche. His group is based at the Institute of Biology, which is part of the École Normale Supérieure in Paris. He has a long-standing interest in DNA methylation and chromatin-based epigenetic processes, and his group, the research of his group, focuses on transmission of chromatin states in Arabidopsis, which is a plant. Next in, with number 2, we have Troy Day. I'm a bit disappointed that you're not doing stretching exercises or warming up, but anyway. Okay, so Troy Day is a professor on Canada Research Chair in Mathematical Biology. He has a dual appointment between the Department of Mathematics and Statistics and the Department of Biology at Queen's University in Kingston, Ontario, in Canada. He's trained as a biologist as an undergraduate, but then did a PhD in mathematics, and so is a proper mathematical biologist. His main research interests are in evolutionary theory, and he has recently been involved in collaborative work developing theory for non-genetic inheritance and evolution. Going on with the letter D, we have Ben Dickens, who is a postdoctoral researcher at the Center for Comparative Genomics and Bioinformatics at Penn State University. He is also a former UCL graduate, so he's homegrown talent. And he uses experimental and computational approaches to study population and molecular genetics. I made that up. I hope it's more or less what you're doing. And finally, we have Eva Jablonka, who is a professor at the Cohn Institute at the University of Tel Aviv. She has been working together with Marion Lamb on the evolutionary implications of epigenetic inheritance for a long time, probably about the time when the current learn crop were born. She started doing that. Okay, so these are our speakers, and I hope I've stuck to time and will now hand over to whoever the first speaker is.
Thank you. Gosh, it is quite loud. If the speakers want to make themselves comfortable, Eva will be the first speaker. What we're going to do before the debate begins is have a vote. So you can all see the motion above me on the slide. Epigenetic inheritance compels an extension of the modern synthesis. You have red and green cards in front of you. If you agree with the motion, then please hold up a green card. And if you disagree with the motion, then please hold up a red card. And what we're going to do is, with the naked eye, get a sense of where people are on this pre-debate, but also one of our committee members is going to attempt to photograph this phenomenon and count it later, and then put it on our website, and do the same after the debate as well. So please hold up your cards. Remember, if you agree with the motion, green. If you disagree, red. Well, you know, the Greens have it at the moment in this room, I'd say. However, Anna is hopefully taking a good photo of this room and can tell us the truth on our website a bit later. Also, I would just like to reiterate one more time before we get started that the event is being filmed, so if you would prefer not to have your face on camera, then can you just stick your hand in the air now and we'll make sure that our cameraman avoids you. Anyone new that would prefer not to be filmed? Okay, we can get started. So the order in which speakers are going to go is Eva Jablonka, Ben Dickens, Troy Day and Vincent Collot for the main presentations. So Eva, I invite you to the podium.
Well, hello and good afternoon, and thank you very, very much for inviting me. I hope not to ruin this very nice vote that I saw, the results, so I'll do my best. Okay, so I'm going to talk for the motion, epigenetic inheritance compels an extension of the modern synthesis. So what is the main argument that I'm going to present to you? The main argument is this. We all accept that heredity is a fundamental property of living organisms, and the changes in our understanding of heredity lead to changes in our understanding of evolution, necessarily. I argue that the modern synthesis was based on a limited notion of heredity. Our notion of heredity today, and not only today, it has been expanding during the last 25 years. And it is now including aspects that we didn't think about. It includes, among other things, epigenetic inheritance of what I have just said. I will argue that evolutionary theory has to be extended, it has to be remodernized. It's a very big body of theory. I'm going to focus specifically on the things that are relevant for heredity. Then I'm going to tell you what epigenetic inheritance is, how we define it, what we mean by it, and I'm going to end by saying what difference our understanding of epigenetic inheritance must make, I believe, to the modern synthesis view of evolution. So what are the assumptions of the modern synthesis? And I'm focusing mainly on those that are related to heredity. First, and I'm talking about the 1980s version, which is still seen in most textbooks of evolutionary biology. So the first assumption is that heredity occurs through the transmission of germline genes. There are no inherited variations that cannot be explained in terms of variations in DNA. This is the first assumption. When I'm taking the positive aspect of the assumptions, I'm not saying they are wrong. I'm saying that they are limited. And that the things in red, it is negative aspects that are being challenged. Second assumption is that hereditary variation is the consequence of, first, a combination of pre-existing alleles that are generated by the sexual processes, and two, new variations, mutations, that are the result of accidental changes in DNA. The negative aspect, which is being challenged, is that hereditary variation is not affected by the developmental history of the individual. There is no soft inheritance. I will explain what soft inheritance means exactly in a few minutes. The third assumption is that heritable variations have small effects, and evolution is typically gradual. Through the selection of individuals with phenotypes that make them slightly more adapted to their environment than other individuals are, some alleles become more numerous in the population. Mutation pressure or variation pressure is not an important factor in evolution. With a few exceptions, macroevolution is continuous with microevolution and does not require any extra considerations beyond those operating during microevolution. So macroevolution is just more of the same. There are other, I will touch these three first assumptions during my talk. The others that I'm enumerating here are also a very important part of the modern synthesis. I will not talk about them. But all of them, all the things in red, are being challenged. So I'll just say what they are very, very briefly. The fourth is that morphological evolutionary innovations are the results of cumulative gene mutations. Generic physical chemical processes are merely a constraint specifying the boundaries of physically possible forms. Plasticity, in the negative sense, is, in the negative way of putting it, is not a generative cause of morphology. The fifth is something that you have been talking about last year, and I understand you voted against, that the targets of selections are individuals, although conspecifics in group interact and may co-evolve with each other, as well as with their symbionts and parasites. Group selection or community selection is rare. The community is rarely considered as a target of selection. It's not just group selection in the terms of groups of, let's say, human beings or mice or rats or whatever. It's also the community in terms of the individual as a community of genomes that co-evolve together, we and our parasites. But the assumption is that the community is rarely considered as a target of selection. Again, there is quite a strong challenge to that. 6, evolution occurs through modification from a common ancestor and is based on vertical descent. Horizontal gene transfer has minor significance. It does not alter the basic branching structure of phylogenetic divergence. Again, the negative, which is being challenged, the main pattern of evolutionary divergence at all times and for all taxa is tree-like, not web-like. If you will look at the new issue of Biology and Philosophy, it is all devoted to this point, and it is very strongly challenged, especially with respect to bacteria, fungi, and plants, but not only. The bottom line of this view of evolution is that the only direction-giving process in evolution is natural selection. This is the important thing. Now, I want to present, you probably all know this gentleman. This is Ernst Mayr, one of the architects of the modern synthesis, one of its strong advocates and popularizers. He did a lot to establish the modern synthesis as the version of evolutionary theory that dominated, I think, evolutionary thinking for the last 60 years. Now, what does he say about the modern synthesis? So that's Mayr's view. He says, at a symposium at Princeton in 1947, representatives of the most diverse branches of biology, including ecology, came together and declared their acceptance of this newly synthesized integrated evolutionary theory. Only the developmental biologists stayed outside, insisting that the Darwinian theory was unable to provide an explanation of the evolutionary phenomena encountered by them. Developmental biology, by most accounts of the modern synthesis, was left out. And it is, of course, epigenetic inheritance is an aspect of development, so it is not surprising that phenomena that had something to do with the kind of things that I'll be talking about were also left out of the synthesis. And he said also, he repeated this many times, also in 2002, 3 years before his death, he more or less repeated the same kind of argument. He said that it was perhaps the greatest contribution of the young science of genetics to show that soft inheritance does not exist. Now, what is soft inheritance? But before that, this is the Princeton conference where the modern synthesis was established, the very famous conference in 1947. As you see, it is rather sexually monomorphic. As Mariam has suggested, the women probably made the tea. But this is what Mayr's evolutionary synthesis is suggesting. There is the evolutionary synthesis in which we have inputs from many branches of biology. Population genetics is very, very fundamental to it, and the assumptions on which population genetics is based are Mendelian assumptions and neo-Darwinian assumptions. That is, assumption that the gene is not changed by the developmental history of the individual or its ancestors. And what is left out is embryology. Now, soft inheritance. So I repeat, he said it was perhaps the greatest contribution of the young science of genetics to show that soft inheritance does not exist. What is soft inheritance? So here is what Mayr says. I use this term to designate the belief in a gradual change of the hereditary material. Put hereditary instead of genetic. He used genetic. The hereditary material itself, either by use or disuse, or by some internal progressive tendencies, or through the direct effect of the environment. Now, it's important to note here that the hereditary change need not be adaptive. It does have to have some specificity, however, for this to be valid. So again, the fundamental assumption of the modern synthesis is the only direction-giving process in evolution is natural selection. All heritable and one of the things that it means many things, but one of the things that interests us here is that all heritable variations are non-directed, and developmental variation, the result of use and disuse and direct environmental effects, are not hereditary. Okay, now there were all the time, throughout the whole 20th century, there were data that did not fit with the modern synthesis. And this data were ignored, and they were ignored for many reasons, both reasons that are internal to the development of genetics and biology, and reasons that are external, political reasons, and so on. But here is what Lindegren, a very great geneticist, said in 1949, that genetical data on which the modern conception of the gene is based are intensely selected data. He was working on Neurospora, and he said that the two-thirds of the mutations that he discovers are non-Mendelian. But this two-thirds, 66%, are simply usually thrown to the bin because they don't know what to do with it. And Nami, who was studying Paramecium, said in 1957, he said that later too, it is perhaps only natural that investigations of messy characteristics are discontinued before publication and that investigators move on to traits more readily analyzed. And I must say that there is a lot of logic in it. If you don't have a model and if you don't have a mechanism for understanding the phenomena that you are seeing, what can you do with them? There isn't very much that you can do with them. You move to think that you can understand, that you have some kind of theoretical framework for working with them, for doing experiments with the beasts and so on, or the plants. So, today, however, development is reintroduced into evolutionary biology, and in addition to many other things, again, that are happening about which I will not talk, the developmental aspects of heredity are being considered. And what I must say here, and this is the second part, is that heredity is not equivalent to inheritance of DNA variation. The notion of heredity is broader than that. So there are different modes of inheritance. We can think about inheritance through the transmission of DNA variation, which can be transmitted in sexually reproducing organisms through the germline. We can think about the transmission of epigenetic cellular variations, which can also be transmitted through the germline, although we know very little about how this happens. We can think about transmission through the reconstruction of early development, and that would not require transmission through the germline, of anything through the germline. It can be a developmental reconstruction, what Marianne and I call soma-to-soma transmission. We can transmit information from one generation to the next through socially learned behavior and reconstruction of later development and ecology, again, soma-to-soma, and in our species through symbolic culture. I will focus, since this motion is about epigenetic inheritance, I'll focus on epigenetic inheritance, mainly two, and a little bit mainly two. Now epigenetics, we heard what epigenetics is. There is quite a lot of confusion about what this term means, so I want to define it for you as I'm using it. Epigenetics is the study in both prokaryotes and eukaryotes of the processes that can lead to long-term persistent developmental effects. At the cellular level, in sexually reproducing organisms, these are the processes involved in cell determination and differentiation. At higher levels of biological organization, epigenetic mechanisms underlie self-sustaining interaction between groups of cells that lead to physiological, behavioral, and morphological persistence. So it's persistence. It's those mechanisms that are responsible for persistence of certain variant states for a long time. And epigenetic inheritance is a component of epigenetics. It occurs when phenotypic variations that do not stem from variations in DNA-based sequences are transmitted to subsequent generations of cells or organisms. And this word, epigenetic inheritance, is also used in a broad sense and in a narrow sense. And there is, again, quite a lot of confusion because of that in the literature. So again, I will just say in a few words what I mean. So, Epigenetic inheritance systems, I will start with the mechanisms. The systems are the mechanisms and the factors that underlie the transmission of functional and structural non-DNA sequence variations. And the cellular mechanisms we have in mind are the mechanisms that involve self-sustaining loops, all kinds of regulatory biochemical circuits that usually involve positive feedback, structural three-dimensional templating, for example, the kind of retemplating, reformation, reproduction of the same structure that we see in prions and in cortical inheritance in paramecia. Chromatin marking systems, these include many things. They include DNA methylation, histone modifications, many types, and non-histone protein marks, and RNA-mediated inheritance, which includes siRNAs, microRNAs, piRNAs, a whole family of little RNAs. And the chromatin marking and the RNA-mediated inheritance systems interact usually. Not necessarily, but usually, as far as we know. There are also whole organism mechanisms, and we can think about the organismal epigenetic inheritance, which involves reconstruction, for example, of maternal hormonal profiles, and I can give examples, but there won't be time. Behavioral inheritance, for example, through social learning, and what is happening on this occasion, cultural symbol-mediated transmission of information. So I'm going to focus on the cellular mechanisms. And in a survey that was published 1 year ago, a colleague of mine and myself, we surveyed the literature for a long time, and we found over 100 cases of epigenetic inheritance, cellular epigenetic inheritance in 42 species. Now, the 102 cases involve, in many cases, one is looking at many genes and many loci that have some kind of epigenetic modifications, and these modifications seem to be transmitted between generations. Again, in many cases, we don't know the exact mechanism. There's a lot to be discovered about this. This is what we see. So I think that we cannot say anymore that we have just anecdotal evidence for epigenetic inheritance. Epigenetic inheritance happens, and in fact, one of the people who is talking against the motion, Vincent Collot, has done some of the greatest work on showing us that in Arabidopsis, quite a lot of genomically induced epigenetic variations can be inherited for many generations. Not all, but there is quite a noticeable fraction of the genome. And since we published this review, there are many more examples, and they're cropping up all the time. So I want also to make a note, because we define epigenetic inheritance as the inheritance of non-DNA variations. However, the epigenetic control mechanisms that underlie epigenetic inheritance, the inheritance of non-DNA variations, are involved in the generation of developmental variation in DNA that can be inherited between generations. And this is something very important, because although we concentrate on the effect, we concentrate here and we can concentrate during evolution for a short time on variations that happen at the epigenetic level. The epigenetic level can affect and is affected by the genetic level, and the two interact. So if something happens at the epigenetic level, it can lead to all kinds of genetic effects, which can be very interesting and very important. Now, what are the implications of these things for evolutionary studies? What do we need to change? So, first of all, there can be repeated induction of epigenetic variation. And this can drive evolutionary change. First, when there is an environmental change that changes the epigenetic profile of individuals, it can affect many individuals, and it can happen again and again. So most of the population can be affected. And adaptation can occur through the selection of induced heritable epialleles without any genetic change. If we have a lot of epigenetic variation, new epigenetic variation that are induced by some change in the environment, some of them can affect the population in a way that is advantageous for the organisms, and this will be selected. And the point is that it is not just general epigenetic variation that can be induced, but they can have some specificity with relation, which is correlated with the environmental change. Then the epigenetic control mechanism can underlie macroevolutionary changes, such as those, for example, that follow genetic exchanges among different species. Epigenetic differences can sometimes initiate reproductive isolation and lead to speciation, or they can make it more likely that speciation will occur. These 3 points, I will give you a few examples. The others I will not.
Your time's up, Professor Jablonka, I'm afraid.
Oh, you didn't? Did you show me?
Yes, we thought you saw them.
No, okay, can I have another minute?
Yes, you can.
Okay.
We'll have to give everyone another minute. Sorry.
Okay, so the first point was that unlike genetic mutation, the same epigenetic variation can be induced repeatedly and affect most members of the population. This is one example. This is the flax example. This is a plant which has been, the fertilizer has been changed. As a result, there is a very big change between the wild type on the left and the induced one on the right, and this is inherited for 6 generations. The change is both genetic and epigenetic. There is another example of androgen suppressors. The induction is by an androgen suppressor, and it is transmitted through the male line for at least 3 generations, some things for 6. Selection of induced and accidental epigenetic variation can occur, and we have several examples of this, one in bacteria and one in yeast. Speciation and macroevolution variation in evolution, the epigenetic effect of stress. Here we have hybridization. We can reconstruct the wheat by doing two hybridizations, and this hybridization lead to a reconstruction of domestic wheat, and they involve huge, massive changes in the epigenome, which are inherited. 13% of the methylation profile is changed. Now, so this is the summary. Epigenetic heritable developmental variation can be induced in sexually and asexually reproducing organisms. The same range of heritable variation can be induced repeatedly. Stress-induced developmental variation can be extensive and include many loci. And this has implications for evolution. It can increase the rate of adaptive evolution through natural selection, generate non-adaptive evolutionary trends, contribute to macroevolution and to speciation. So this is the bottom line. Epigenetic inheritance challenges some of the neo-Darwinian assumptions of the modern synthesis. Therefore, the modern synthesis need to be modernized. Thank you very much.